Fine. So if we see now here, this is showing the size of the conceptus. Fertilization हो गया, अब वो implant हो रहा है. Actual size is this in the beginning. If that is cut and seen, then you can see it is forming this conceptus, where this is outer cell mass, this is inner cell mass. So initially जो हमने देखा था वो conceptus single cell with two set of nucleus, then cleavage, then two cells proper, 3, 4, 6, 7, 8, 16, mulberry shaped conceptus, solid, and then which is called as morula, and then there is a cavitation inside. छोटे छोटे कैविटेशन बनते हैं और ये ब्लास्टोसिस कैविटी बनता है और उसके बाद इस कैविटेशन के कारण एक सेल अंदर के साइड रहता है which is called as inner cell mass, एक outer cell में साइड रहता है which is called as outer cell mass. Outer cell mass again divides into two. Inner is cytotrophoblast and outer is in situ trophoblast. Inner cell mass is divided into epiblast and hypoblast. और जैसे जैसे बड़ा होता है वैसे वैसे epiblastic cell gives rise to another covering cell called as amniogenic cell and there is a small amniotic cavity formed. अब ये amniotic cavity बड़ी होती जाएगी, ये छोटा होता जाएगा. So you can see the amniotic cavity is increasing in size and this is decreasing in size. Blastocyst cavity original, which is now called as yolk sac, and the cells of the hypoblast, they are pushing towards here to form a covering, and that covering is called as the Users membrane. 1 minute, just a minute. Now moving further, if we see, so this is day 9. Parallelly, if we are talking about it, this is day 9. There is this amniotic cavity increasing in size. Changes are happening in this inner cell mass. Changes are also happening in this outer cell mass. And as you correctly must be knowing, this outer cell mass is going to form trophoblast, which is divided into cytotrophoblast and outside trophoblast, and there are small cavitations in it. This is going to form placenta. And these epiblast and hypoblast are going to form the double, this epiblast, not actual hypoblast, this hypoblast will actually form just the layers of this yolk sac, ultimately Husser's membrane, but this epiblast is going to form all the three embryonic layers of the body: ectoderm, mesoderm, and endoderm. Well, so if we see it further, now here you see these cells are changing dimension, this is increasing in size, this is primary yolk sac, hypoblast is there. Now further going, there is a layer formed outside, and this outside is layer is called as, which is outside the inner cell mass. So it is called as extra embryonic mesoderm. This is extra embryonic mesoderm, which is being formed from the outer cytotrophoblastic layer as well as maybe some contribution from the epiblast. So this is extra embryonic mesoderm. And then small cavities appearing, and then it is becoming smaller and smaller in size. And then if you see, this is definitive yolk sac, and we have the whole Not only that, not only definitive York sac, but also in the extra-embryonic mesoderm, small cavities are appearing, which will be uniting and forming another cavity. And because of that, this will be extra-embryonic splanchnopluric mesoderm. This will be extra-embryonic somatoplurid mesoderm. यहाँ भी कैविटी हो रही है, आउटसाइड में ट्रोफोब्लास्ट में भी कैविटी हो रही. You can see the size has increased now to this size. Still it is so small in size. Actual size is this. What is being seen here is a diagrammatic representation of an enlarged view. Definitive York sac. And if you see, because of this germ line, now we had seen the epiblast starts proliferating, and in the lower part of the embryo, if this is the upper part of the outer side of the embryo, then this is amniotic cavity. और उसको हम ऊपर से देख रहे हैं. So this gray colored thing is epiblast. This blue colored thing beneath it will be hypoblast. But this epiblast initially in the lower part will form primitive streak. From the primitive streak only will come mesoderm, and this mesoderm is inside the embryo, so it is intra-embryonic mesoderm. वो सबसे पहला अगर अपन इसमें देखें, so epiblast के cells जो layer बनाते हैं, now if you see this, if I cut or if we cut this and see what is visible, then the thing visible will be like this. This is the gray color thing, this is blue color thing, this is hypoblast. Now this epiblast will first of all form endoderm. The cells of epiblast will form and will come to replace the hypoblast. So epiblast first cells derived from the epiblast are endodermal cells. They replace the hypoblast. You can see it happens within 14 to 15 days. Then during 16th day, the primitive streak जो हम बात कर रहे हैं वो बनता है, and the cells of them will form intra-embryonic mesoderm, and they will pass between the overlying epiblast and underlying endoderm, and they will go everywhere. As you can see, this is primitive streak. It is forming intra-embryonic mesoderm. It will go everywhere between the overlying epiblast and underlying endoderm. Overlying epiblast, as you can see, underlying endoderm. साइड में, ऊपर, नीचे, except at two places जो हमने देखा था, we know. One is cranially, which is called as cloacaudal plate. Second is caudally, which is called as cloacal membrane. So you can see they are going it like this everywhere, and this is how they are spread, and the whatever cells which remain of the epiblast, they are called ectoderm, or they become ectoderm. In the center there is notochord, which is also mesoderm only, but the central mesoderm which spreads from the primitive streak till the procaudal plate, that part is called as notochord. So this is notochord. और साइड वाले जो mesoderm है, which is intra-embryonic mesoderm, जो अभी बना है, अपन ने बात किया, सेकंड लेयर, सबसे पहले एंडोडर्म बनता है, फिर mesoderm बनता है, फिर जो बाकी के एपिब्लास्ट सेल्स होते हैं, they get converted into ectoderm. So mesoderm जो होता है वो तीन में डिवाइड हो जाता है. यहाँ पे जो mesoderm दिख रहा है आपको, ये सबसे मीडियल जो होगा पैराक्सियल mesoderm, सबसे lateral is lateral plate mesoderm, in between will be intermediate plate mesoderm. So it will be like this. If you see, यहाँ पे लाइट नहीं है, it's become very hot. Just a minute. Okay, so we know that these are the three layers which are formed. If you see, this is mesoderm. So paraaxial, intermediate plate, and lateral plate. Lateral plate again divided into splanchnopluric and somatoplurid. This paraaxial mesoderm is going to form somites. Days बढ़ रहे हैं, 17 days, 19 days. So this paraaxial mesoderm is going to form somites. Primitive streak, as we know, is the mother of intra-embryonic mesoderm. It forms paraaxial mesoderm, intermediate mesoderm, lateral plate mesoderm, Like this, you can see paraxial, intermediate, lateral plate. Paraxial, and then the post-aortic, the paraxial mesoderm is going to form somite. ये सब आपको बताया था. उसके अंदर पोस्ट ऑटिक पार्ट होता है और पोस्ट ऑटिक पार्ट में सेगमेंटेशन होता है in cranio-caudal direction, and the segmentation forms somites. They appear between 28 to 30 days, at the end of which about 40 full pair of somites are laid down in the human. पैराक्सियल मेसोडर्म पूछे तो भी वही लिखना है. सोमाइट मतलब तो सोमाइट्स के बारे में लिखना है. सोमाइट्स पूछे तो भी सोमाइट के बारे में लिखना है. और इंट्रा एम्ब्रियोनिक मेसोडर्म पूछेगा तो भी अभी तो तुमको पता है वो मेनली पैराक्सियल मेसोडर्म पता है. So it will come boil down to mainly somite. So post-aortic part becomes segmented from somites. Rostral to the aortic vesicle, it remains unsegmented. It forms, it underlies a developing brain, and it develops base of the skull and all striatal muscles of the head. So here, if you see the somites, 44 pairs are formed. Then the caudal most 7 disappear. So ultimately, जो बचते हैं वो 37 हैं. 1 to 4 are occipital. They form occipital part of the skull, bones of the nose and muscle of the tongue, and muscle of the tongue, bones of nose and eyes. As you can see, cervical next 8 pair it forms in the presumptive cervical region, give rise to occipital bone, cervical vertebrae, and associated muscle. Thoracic is next 12 pairs. It forms thoracic somites, thoracic vertebra, and associated muscles, and 5 lumbar somites and 5 sacral somites. Finally, 3 coxal somites disappear. Finally, 3 coxal somites are there. 7 coxal somites disappear. So this is how the somites are formed from the paraxial mesoderm. Number of somites. They give a good index of age of the embryo. First somite appears on approximately 20th day, and roughly 3 somites are added each day. Roughly 3 somites are added each day, as you can see. And in the somites, if we see how do they develop, so first it is myocele. If you see here, this diagram, this is a somite. Then what happens to it is such cavitation develops in between, and there's a medial layer and then there's an outer layer. Medial layer is going to form the vertebral column. The outer layer is going to form dermatome and myotome. So presumptive vertebral body is formed medially, and later it is dermatome and myotome. Initially, the somites contain a cavity, myocele, which is later obliterated. Somites may, if we see, there is a ventromedial sclerotome, dorsolateral dermatomyotome. ये याद रखना है. कैविटी बनती है, मायोसिल, कैविटी बाद में खत्म हो जाती है. तो ventromedial part जो है, it is called as sclerotome, which is this. It is surrounding the developing neural tube. Dorsal part is dermatomyotome. Cells of the sclerotome, which is medial, lose epithelial character, become polymorphous, and migrate ventromedially around the notochord. This is notochord, this is neural tube, and they form primitive vertebra. Dermatomyotome is a lateral part. It consists of lateral dermal plate. It forms dermis and subcutaneous tissue. मैंने बताया था आपको लेक्चर में यहाँ पे. Epidermis is formed from the ectoderm. This is mesoderm. We are talking about mesoderm. So mesoderm forms dermis and subcutaneous tissue. Medial muscle plate forms skeletal muscle. So this is medial muscle plate, myotome. Myotome will form skeletal muscle of the region. Each mesodermal somite will have corresponding spinal nerve. Muscle will migrate, but with its nerve supply. And this is grossly what we saw about the somites. Now I end here and I ask you if there is any question because